Interesting talk that's going to be given by Mrs. Mitra Cowan. I have to tell everyone that our original plan, if we had been doing this in a conference room, was that Mitra was actually going to be moving micro-injection microscopes into the room where the conference was going to be held. And those of you that would have wanted to test out micro-injecting constructs into blastocysts would have had this opportunity on site. Of course, COVID threw a monkey wrench into our organization, and we have not yet found a way to do this on Zoom, unless she's got a video that she's going to be showing us, which is, of course, interesting. You'll get to see it, but maybe you won't get to test it yourself. So we'll see what life produces as we go on. Mitra is the Associate Director of the McGill Integrated Core for Animal Modeling, which stands for the MICAM facility. Mitra actually studied in reproductive biology and embryonic stem cell technologies some years ago at the University of Illinois in the late 1990s. We won't tell how long. She took up a position at the Johns Hopkins in the United States, and where she was managing the transgenic core facility in the for some time. Then she followed the love of her life and moved to Montreal, snowy Montreal. And she basically started up a new transgenic core at the Centre de Recherche du Centre Hospitalier de l'Université de Montréal. She was there for a number of years and set up an amazing facility from 2004 to 2017. And then we actually managed to steal her away and bring us to work with us at the GCRC. She's been there since 2017 and has essentially revolutionized a lot of the technology that we have been doing, and she is essentially able to service a number of people within McGill, but also many different facilities outside McGill. And those of you interested in knowing more about this are invited to contact Mitra. We'll put her coordinates maybe in the chat so that you know how to reach her. And she's now generated over 150 different mouse models. So tell us all about, maybe not the 150, but some of them.
Okay, so
you need to share your screen.
Share my screen. And I'm assuming you can see this?
Yes, perfectly.
Perfect. All right. So if you don't mind, I'm not going to look at myself and just look at my screen.
Okay.
So first, thanks for the kind introduction, and it's a pleasure to be here, and I am excited to be able to share some of our experiences and our expertise to help the participants potentially generate their own genetically engineered mouse model. And I think hopefully you've been convinced by Yojiro and Bill Muller, who has been doing this for decades, the value in using mouse models to study cancer and to answer kind of the research questions that you might have. So just to give you a brief overview of the platform, as Nicole introduced, we're the McGill Integrated Core for Animal Modeling. The idea being that I think part of the reason I joined is because I have to say the Cancer Center and McGill were making some of the most interesting models, and they were really interested in staying on the forefront of technology. And since I'd been in the field for a long time, I enjoyed, you know, developing the newest and most interesting models. I also think the demand at McGill was to grow the core, to share their expertise, because I think other researchers wanted access. So basically, our core has grown to be able to service all of the McGill University and its affiliated research institutes and hospitals, including the M&I, the Lady Davis Jewish, Douglas, as well as all the research center at the RIMUHC. So basically, our mandate is to be the platform to create all the new genetically modified animals, as well as offer support services to help researchers manage their lines after they've been developed. So today, obviously, I'm going to spend my time talking about using CRISPR-Cas9 technology to create mouse models. But I do want to mention I have added to my protocol, and we are starting to do some rat models here as well. So a lot of well, actually everything that I've spoken to is applicable also in rat. And so the idea is that, we can use this technology in other rodent models. And then of course, we're still offering all of our classic services that the lab has well established over the years. I think we've had a lot of discussion and introduction of CRISPR-Cas9. I think Yojiro did an excellent job of this, so I don't really want to spend a lot of time on this, but of course our goal was really to use the CRISPR-Cas9 technology and how we were going to deliver these components through microinjection, and we actually now have a couple other delivery mechanisms to create the double-stranded repair and either use NHEJ, non-homologous end joining, or using a donor template to introduce some kind of targeted mutation. So the question everybody asks is what kind of mouse models that I can make, and I think you've seen a few of these already. So what I thought I would put up here is a slide of all of the confirmed CRISPR models that we have made since I joined in 2017. For the last 2.5 years, we've generated over 100 different confirmed CRISPR models. As you can see here, they range from the straightforward, from knockouts using NHEJ, which we also have generated some by exon deletions, to more complex models. And actually, most of the models that we have created are using homologous directed repair, or actually the introduction of a donor template to introduce a point mutation. We have generated flags, obviously conditional mice, which is the introduction of LOXP sites. And actually, I would say the largest and newest demand is the ability to be able to generate large knock-ins. CRISPR-Cas9 technology is continually developing. And so this is an expertise that I think, especially with Yojiro, has been able to introduce in our lab. And as you can see, this is now our most second popular kind of model we generate. But I'm hoping to be able to show with you that because we've generated over 100 models using CRISPR-Cas9 in the last couple of years, that we can share not only our expertise, but kind of the best practices that we've developed and how you can utilize them in generating your own mouse models. So the next thing I'd like to kind of introduce is the idea of timelines. Everybody's very excited about the experiments that they might be able to do with their new genetically engineered mouse model, but it's important to kind of think about how long it's just going to take to generate the mouse of itself. And it's kind of follows the idea of the easier the types of models they are, the kind of where we're just using NHEJ, straightforward knockouts, which are the indels or exon deletions. These are the shortest, of course, because all they are is the introduction of the guide and Cas9 into the delivery within after microinjection, pups are born, and then the screening. Anytime you get to introduction of some kind of homologous directed repair where there's a donor template involved, that obviously becomes more complex. Those steps that would be involved is designing a donor template. Nowadays, I'll speak about this, is we pretty much have all our donor templates generated synthetically, the timeline to receive that, microinjection or delivering that template, as well as then the founder mouse screening. I will actually say the knock-ins and point mutations, small insertions, they're fairly still straightforward. We've really developed some good protocols. We usually have about 25% of the mice born or potential founders. Those are fairly straightforward and the timeline isn't much longer, but the large knock-ins and especially conditional alleles are still the most complex. And not only is it in designing donor templates, but even screening the mice is quite complex still today. So they do take more time to make. So these are just to give you an idea of general timelines. Obviously things can take longer if the targeting region is more complex, there are more variants, but it's just to give you kind of something to plan for. So now that I've kind of shown you kind of what kinds of models we can make, some timelines, let's just dive straight in. I'm just going to talk about kind of the key steps that we need to think about when we generate your new genetically engineered mouse model. And there's no surprises here, you know. I think you can guess what they would be. Obviously, the first step is you have to design it, and either you can work on this, and I'm going to try to give you some of our best practices and some ideas to think about, but obviously we're here to help you at any time. And even you can design it and then we can go over any of your designs. But after you design it, then obviously then the next step is kind of choosing the appropriate guide RNAs and then testing the cutting efficiency. You'll see why this is very important because as you saw in my earlier slide, with generating over 100 mouse models using CRISPR, actually, if you notice, I think over 90 of the 111 were using some kind of donor template. So it's a very important that we have the best guides that we possibly could have and the highest cutting efficiency, because if we're going to introduce a donor template, we want to make sure those guides cut, because otherwise the donor template will not be incorporated. After we have chosen our guides, we then obviously have to design the donor template based on the chosen guide. I will talk about actually the delivering of CRISPR components. And we actually have 3 different delivery methods that I'll talk about. And maybe someday we can re-give this course in the future by both microinjection, we use electroporation, and Yojiro has introduced the idea of kind of iGonad as kind of this additional delivery method directly into the mice. So then I will also touch some ideas about how to screen your founder mice, as well as I want to just talk about briefly about breeding your founder mice so that you won't have any problems in the long term. So let's dive straight into actually designing your model. Basically, for us, there are basically 2 kind of designs. We kind of classified into 2 different groups. The 2 different groups that we have are NHE-mediated alleles. Those are kind of fairly straightforward. They're generating some kind of functional null allele by an indel mutation or by a deletion of a single or several exons, or we have HDR-mediated alleles. And a lot of the key practices that we use, our best practices, are very similar, but we really kind of classify that in one of the 2 groups. So then, oops, next slide. Okay, so, you know, the things that you kind of have to think about when you're doing NHEJ mediated allele, the design things of obviously is what you have to think about in these is how many guides are you going to use. You can either use 1 guide or multiple guides. There's good strategies for doing both. How many targets are you going to potentially do? I'm going to talk a little bit later for one of our projects that we've worked on with Dr. Maxime Bouchard's lab, where we actually targeted multiple genes. So the idea is that you can target multiple locuses using NHEJ, and some of the potential mice you could generate that way. And then obviously when it comes to HDR mediated allele, it's really, you have to think about what type of allele are you going to create. And there's quite a large variety of those alleles, from you've seen already point mutations to the conditional to the large insertions. But the real point of this slide is the third point. When you're designing your model and what you want to get inserted, you need to think about your long-term genotyping strategy now. This I cannot stress enough. There's nothing worse than having, I have seen it, where people have ended up using sequencing as the only way to genotype a mouse. That is not a long-term genotyping strategy. If you think about it now and you incorporate some kind of PCR strategy, either using restriction sites, other cloning strategies, you need to incorporate this in the design for, it doesn't matter which model you're making, NHEJ or HDR. These things have to be thought about now. And I like to stress about this because people don't always incorporate this in the design. And the last thing I want to say is that there is no one size fits all. A lot of alleles have very specific unique variants, and we like to think carefully about each individual design. I will not say, okay, just always do this if you have a point mutation. If you make a design, make your point mutation, we will look at it and we'll still look, is that going to be the best strategy? Is that actually going to get you the model that you want? And are you going to be able to genotype that in the future? This allows us to avoid future complications. Do your front end work so you avoid back end work, because back end work is way harder and more complicated in the end. So again, I'm going to just kind of go into the designs of the two different types of models. The first one being NHEJ. Obviously, as I've talked about, and you've had already introduced the idea of using some kind of indel, or so that you kind of have a premature stop codon, or you can have some kind of exon deletion. And then I just wanted to put a figure here is that some people are using CRISPR-Cas9 to actually make large deletions, huge, actually 65 kb. There are reports. Our lab has personally never actually done a model like that, but there are reports out there, so the possibility is there, but there are a lot of challenges with these types of models. But we are much more experienced making exon deletions where we're flanking either a single exon or multiple exons with guides. So these are actually fairly easy. Once you identify the target sequence, you're just going to search for the guides that you would potentially want to use using one of the online CRISPR tools. We use CRISPR, that's our tool of preference. There are others out there, but we've been using this tool for over 2 years. And when I talk to other transgenic labs, this is kind of the tool of choice. I've put the link here for you, and I'll show you some kind of outputs that they would give us a little bit later when I show you how we choose guides. But when we're doing our NHE-mediated allele designs, there are a couple things I want to talk about. It's a super easy kind of design to make because you just kind of choose the target, you pump that sequence into the online tool, it's going to spit out the guide, you microinject it, it's really fast and easy. It holds a lot of appeal because people think they're going to get their mice very quickly. I think actually Yujiro brought up this point already. They can be a challenge to genotype. I cannot predict the repair that's going to be generated by microinjection of a single or multiple guides. Sometimes, yes, as people, you get the predicted small single or small deletion or insertion of a couple base pairs, but you can have larger fragments. When you have the larger fragments, you can ablate primer sequences and you will have a hard time identifying the mutations in the mice. The second point I want to make about the con of NHEJ-mediated allele designs is that we have actually noticed that sometimes you get less than ideal F0 or founder mice produced versus the total number of mice with mutations. So what am I trying to actually say with that statement? And I actually thought I'd put a couple little tables that our CRISPR specialist, our RA, Jade Desjardins, presented recently at a transgenic meeting last month in October. And so here I've listed 5 different projects. So they kind of span this, classic NHEJ design where either we were trying to delete an exon spanning with, we were putting 2 guides flanking the exon section. So if you can see that we were trying to span, a 1.7 kb deletion. Here is 1.3 kb deletion that we were trying to get. We actually had pups born, but we couldn't actually figure out if we had any potential founders because we were unable to screen these mice. We're unable to actually screen these mice because we had because of the large deletions, there was so much disruption in the primer sequences, we couldn't actually identify which were the best founders to breed with these for these target mice. And then here, actually, in the last three, you can see here we're trying to make indels with a single guide RNA. We had a reasonable number of pups, and actually we had up to pretty much 50 to 80% of the pups that were born had some kind of mutation that were actually identified and confirmed by Sanger sequencing. But actually, how many of those pups that had that were confirmed with mutations by Sanger sequencing? And here is 0 actually were potential founders. They had no kind of deletion of a single base pair or 2 base pair deletion or insertion. Everything was in frame afterwards. So all 9 of these pups, even though they had mutations, were in frame and did not create kind of a frame shift or a stop codon. And it was the same in this situation. In here, we had 10 pups with potential mutations, but only 1 out of 10 actually was a useful founder for this researcher. So one thing to think about, even though NHEJ-mediated alleles are easy to design, there can be the cons of either the challenge of genotyping, and so then we can't identify potential founders, or the fact that even though we will generate mice with mutation, we actually don't produce founder mice. So this, for us, is kind of a bit frustrating, not only for our lab because we do all this work, but we find it very frustrating to not be able to deliver what the PI wants. So then we started working to actually add a donor template with the designs. And so I just wanted to show you here is that when we added a donor template to actually create either the deletion or introduce the stop codon or the frame shift, here you can see the number of pups that were born. Here you can see the actual number of mice with mutations if we screen for it. But out of the number of pups that were born, you can see that here are all the HDR donor template was actually introduced. So here we have anywhere from 25 to 50% from the pups were born were actually generated potential founder mice. So I think if you're going to make a null allele, you really have to think about the design and what are you going to get at the end so that you can actually have useful founders that you're going to use. And so kind of what I've done here is kind of introduce the idea that although NHEG mice are easy to design and actually fast and the cheapest type of mice to make, that we want to think more about using HDR because these are going to actually create more useful founder mice for us in the long run. So now that I've introduced the idea of homologous directed repair, I'll just go into those designs. So actually I've kind of already introduced them, the idea of null alleles using HDR, as well as we have small targeted mutagenesis. This includes making point mutations or tags, or introducing a stop codon. And then we also kind of have our larger knock-ins, which are either creating conditional mice or large knock-ins. So again, we kind of break these down into the 2 groups. I've already introduced the idea of how are we going to design using homologous directed repair of a small insertion, and we kind of have 3 strategies. And I've already introduced the idea here is that if we want to create a precise deletion, we want to make an exon deletion of a single or multiple exons. What we're going to do here is we're going to introduce multiple guides, but we're going to introduce a donor template that's going to anneal the 2 cut sites together to create this precise deletion. And then strategy B and strategy C are really very similar to each other. They're going to use a single guide with the Cas9 protein with a donor template to introduce either the frame shift that you want to introduce by adding or removing a nucleotide, or you're going to introduce the point mutation, a tag, or a stop codon. So these are kind of the general strategies that you can use when you're thinking about designing a mouse model if you're making kind of a small repair. So then if you want to go for large knock-ins, I just want to just touch on these briefly. They're using the kind of same strategies, whether using one guide or two guide, but now you have to start thinking about the design for the donor templates ahead of time. Kind of what here is going to make the difference is, what are you trying to insert? Are you trying to insert a small insertion of a small reporter, something like a GFP, a CRE with an iris or a Kozak? These are kind of these smaller insertions. We can use a donor template called a long SSODN. These have kind of maximum length of 2.2, 2.4 kb. If you're trying to insert a small reporter, GFP is around 700 base pairs. It's fairly straightforward to insert. These are a good option because then you'll still have homology arms that you can use for your targeted region, as well as you can use then a double-stranded donor template if you have then a larger insert that you want to introduce. The idea maybe be doing a CRE-ER GFP, which I will show you later on. These can get up to around 5 or 6 kb. So then we like to use a different kind of donor template. We like these because they have a variety of formats. You can have a circular plasmid, PCR fragment, or what we call a TILD fragment, which is a cut circular plasmid. These are really nice because they allow a lot more flexibility, and they allow a lot more flexibility for your homology arms as well as the length for your homology arms. And then the last thing I want to talk about for designs for homologous directed repair, HDR, is conditional mice. There are really two strategies out there. Again, you're going to have a guide RNAs flanking your exon of where you want to introduce your LOXP sites. You're either going to use 2 small donors or you're going to use 1 large donor. Basically, there's pros, there's cons to everything out there. Again, back to strategy A, where you're using 2 small donor templates. They are cheaper to order, and the templates are faster to receive. The challenge with this, and this is something that's very important to consider, is that you're using 2 different guide RNAs and you're using 2 different small donor templates. What you need is for both of these guide RNAs with the LOXP sites to be introduced, and they have to be introduced in cis. They must be introduced on the same allele. They cannot be introduced on separate alleles, otherwise the CRE-LOX system will not work. So then you can look at to strategy B, because you're going to use 1 donor template which has both of the LOXP sites on the same donor template. There are reasons why we would use one versus the other. We will sit down and look at this, but basically you want to look at using strategy A when you have a kind of a larger region to flank with the LOXP sites, because maybe generating a large donor template would be too difficult, or the genomic sequence can be difficult to synthesize or clone. If we're going to do strategy B, it's more popular, and we like to go this way because we like to have a donor template that includes both LOXP sites to avoid the problem of having the LOXP sites in trans versus cis. But we tend to do these with smaller regions that we're able to flank the LOXP sites where they're around 2 kb. So now that I've talked about kind of designs, we're going to go into the next step. And this is where people kind of, it's a little bit more fun. We're actually going to build the model. So we're going to choose the guide. And so the most important thing about choosing guides is on-target cutting efficiency of the guide RNA. That is the first key element for a successful CRISPR-Cas9 project. And I can't stress enough, and you're going to see why, that efficient guides are really important, because what it does is it requires less rounds of microinjection to generate the founder mice. If we don't have an efficient guide, then we have mice that are born with no mutations, and then we have to go back and we have to microinject the Cas9 components and guides again. So basically, choosing guides is fairly straightforward. We like to use Ensemble to analyze our gene of interest, and then obviously we will choose the target region based on multiple multiple factors. These include from project goals to the structure of the coding sequence, as well as you have to look at all the transcript variants. I don't want to go into too much details for every single gene. Is kind of unique, so you need to spend the time and actually look at your gene. Once we know the target region, as I said before, we just put the target region into our online guide selecting tool, CRISPR. That's our tool of choice. We're going to typically enter between 50 to 200 base pairs. You don't need to enter 1 kb because we're not going to target we don't need to know that many different guides. The important point here is you want to choose the guide for testing that it's as close to the desired mutation as possible. We want to look for guides that are within 10 base pairs, excuse me. There's really a lot of good studies that show the more the distance of the guide is, the lower the introduction of the DNA donor template. So I thought I'd do a snapshot of some guide selection when you use the CRISPR tool. When you enter the sequence into CRISPR, what's going to happen is you're going to get an outread at the top of your screen, which is going to look, here's the sequence. And then what you're going to see is all these little 3 base pairs with these 3 dots. So these are the PAM sequence, which are the necessary sequence for the guides to recognize the target. And then these dots here are the actual going to be the longer guide sequences. So if you click on this on these sequences here, you're going to then be directed to the different guide sequences below for you to look at individually. So what are the key points that we look for when we choose a guide? We want to look for guides that have off targets with 3 mismatches or less. And that is going to be found here in the last box here. I've put a picture here for you. So here we want to have the guides that have the least off target. So we like these numbers to be 0, 0, and 0. You can't always find 0, 0, 0, but we try as hard as we can. And the more, at least we like to have 0 here and 0 here. That means this guide, they cannot find the guide matches with the off targets for within the first few base pairs where the guide lays down. So these are ideal. And then we also do look at these scores for on-target efficiencies. However, one thing I want to stress is all of these efficiency scores are guides that have been tested in cell lines. Cell lines are not the same as testing in vitro in zygotes. So we still insist that guides should be tested in zygotes to validate actually their cutting efficiency. And the last point I want to put here is that guides can actually be designed manually. I would actually say one of our own researchers, I think Dr. Peltier, almost always picks his guides manually, and he's very good at it. So I do want to say that if you want to do this manually, you can. You just need to look for the PAM motif, which is the NGG that is closest to your target. The things you need to look for is a 17 or 20 nucleotide sequence, and you want to look for a GC contact percentage that is between 40 and 80%. So here is, as you can see, this is like a project that I've put here for people to see. So here's the sequence we've put in. This is the exon 4. This is a point mutation. We want to enter this point mutation in here. So we entered this sequence in that I showed you previously. And then these are the 3 guides we picked because they are the best guides that we could find that was very close based on the CRISPR output. That was close to the mutation of interest. And see, as you can see here, we've identified the 3 PAMs, and they're all within 10 base pairs of the mutation of interest. So once you've identified the 3 guides that you want, you basically need to, at this point, before you actually start anything, design a genomic PCR assay for actually screening the guide cutting efficiency, okay? Then you're going to order your guides and you're going to validate them. So I don't need to go over every little step because this is recorded and I'd be happy to share these kinds of information. but you do need to design a genomic PCR protocol for screening the guide cutting efficiency. You have to be able to send the PCR for Sanger sequencing so that we can evaluate whether the guides have cut or not. These are some kind of guidelines that we put down. We want PCR products to be between 0.4 kb and 1 kb. It has to encompass the guide RNA site. It is important that there's 100 base pairs between where the primers start and the cut site for reasonable Sanger sequencing. These are some guidelines for GC content for the PCR product, as well as other guidelines. So the last thing now is to be able to stay within the timeline, I'm just going to kind of move along here, is we order all the guides synthetically. Okay, the reason we order guides, synthetic guides versus in vitro transcribed guides is we've been doing this. We switched over about 2 years ago, and we found that the guides that we receive synthetically are much more cost effective. They're very stable. In vitro transcribed guides tends to be less stable, and we have tested them side by side, and we have greater cutting efficiency using synthetic guides. You can order them from a variety of companies. I've listed 3, but all companies that provide them usually tend to be quite good at this now. And they're all within clearly they're within close price range with each other. So how do you validate the guide RNAs? So we validate the guide RNAs in mouse zygotes in the strain of your choice. What we do is we take each guide and we actually microinject it into 10 to 15 embryos. And then these embryos are then cultured to blastocyst stage. They're lysed, the DNA is extracted. Then we do that key PCR or genomic PCR assay. There is a possibility, because there's a low amount of genomic DNA in the blastocyst, that we'll do either a second or a nested PCR to be able to have enough product to be able to send to for Sanger sequencing. One thing I want to note is we run a subset of that, about 4 or 5 microliters of that PCR product on an agarose gel. If you actually see visible shifts on the gel, these are detections of deletions and insertions, and you don't even need to send it for Sanger sequencing. However, you don't always see deletions or insertions if the deletions or the insertions are too small. So then we will send that for Sanger sequencing. The key point here is you need to look at the chromatograms peaks. Do not look at the text sequence. The text sequence will just give you the dominant sequence. You will not be able to see if there's multiple sequences here. This is really important because we see people regularly miss mutations when they're only looking at text sequence versus looking at the chromatogram peaks. Okay, indels and point mutations are obviously are evidence of misrepair, and we use our software of choice to align sequences as Benchling, and would be happy to help you. But there are other people who use ICE from Synthego, and there are other software out there. But basically, we're going to use the best guide RNA selected based on its cutting efficiency and the proximity to the cut site. So if we go back to the example that I gave you earlier, where these are the 3 guides, we did all those steps that I just talked about. Well, here are the results. This is one that Jad did. And so look how close these guides are. All 3 of them, I chose this example specifically to try to explain why we test the guides. If you look, we tested on average, okay, well, this one we only were able to get 7 embryos, but between 7 to 12 embryos for each one. And you can see that she was able, when she analyzed each individual embryo, only 5 out of 12 for guide 1 actually had some kind of indel mutation. Here, almost all of the embryos were cut, 8 out of 11, 73%. Here, we had 7 embryos, all wild type. There was no cutting with guide number 3. So what I'm trying to stress here is that even guides that are very close to each other will have different cutting efficiencies. It is true, a large number of projects, we test 3 guides, they all have around the same cutting efficiency. But you don't want to be the project where you find that you have guides that have a variety of cutting in efficiency, and you randomly pick one guide and say, oh, well, I'll pick guide 1 because technically it should be the closest to my mutation site. But here you can see it's not the best guide. In this case, the best guide to choose was guide number 2. So this is why we do this all the time, and we recommend it. It's a little bit more upfront work. It takes a little bit more time and money, but it ends up saving you work at the end. So now that I've talked about choosing guides, how to test the efficiency, and we've decided on, in that example, guide 2, we have to actually design a donor template. There are some basic strategies that we use. We have based all our best practices on several papers, but a lot of it comes from this paper that I've put as a reference. It's actually, you'll see the same references on AdGene, they tell you to use it. There's a large number of labs, and I think over the years, we've actually demonstrated that the initial data that they published in that paper is a very good, have very good key strategies in designing homology, SSOD and donor templates. So here I've put a photo from the paper, which will just, just to give you a reminder here, here is the Cas9 with the guide down here. This is the PAM site right here. And the double-stranded break always is occurring between the 3rd and the 4th nucleotide 5' upstream of the PAM. So this is where it's cutting. So this is what is happening to the DNA. It's a double-stranded break. And so now what you need to do is try to design a donor template that will get integrated into that break. So what we do always is we do homology arms that should be starting at the cut site. And then the key piece of information here is point number 2 here is the PAM sequence, or what we call the seed sequence, just the sequence right before the PAM, has to be mutated. This has to be mutated because if it's not mutated, the CRISPR-Cas9 will come in and recut the donor template, okay? So we introduce a mutation here in our donor template. The donor template is always designed as a reverse complement to the PAM strand sequence. And then here for smaller donors, we kind of always do this asymmetric design. And this is based on this paper. It's 36 base pairs on the PAM distal side and 91 base pairs on the PAM proximal side. And this is an example. I just put as a point down here, point 4, the only exception to this is when we design donor templates for deletion templates, we actually just design the 200 base pair homology arms adjacent to the predicted cut sites. And that's the exception for our small donor templates. Here, I just put some examples of, here's the guide that we wanted. And here you can see this is the PAM, and this is where the cut site is going to be. And in this case, the researcher wanted us to create a deletion, a 2 base pair frame shift in here. So here you can see, unfortunately, this PAM, I put this as an example because it's a proline. The problem with prolines is they're always NGG, and that's what the PAM sequence is. This PAM needed to stay in frame. If it is a proline, you can never mutate it. You must keep it. Otherwise, it does not become a proline. Some people talk about changing the amino acid. This is not something that we ever like to do. What works quite well is to actually mutate upstream in the seed sequence, and just where the guide is just about to lay down. And if you make a couple small mutations there, then you can pretty much ablate the ability for the guide and to recognize the target sequence, and then it will not create a double-stranded break after you have your targeted mutation of interest. And then this is a more classic example where what we have done is here's the guide RNA, and you can see here's the point mutation, and this that we've created in the PAM sequence to make it unrecognizable to this guide down here. So now I'll just briefly go over design ideas and points for donor templates for conditional alleles. When you're designing, when you're designing for conditional alleles, if you remember, there were two types of templates. If you're designing a small SSODN to introduce the LOXP site is only 34 base pairs that easily fit inside of small donor templates that range about 200 base pairs. They really kind of follow the strategy I talked about earlier. The key point is to remember, for LOXP sites, we always try to make sure that they are at least 100 base pair away from the closest exon. What we do with LOXP sites is that instead of actually mutating the PAM, is we use the LOXP site to split the guide into two parts. We are able to do this is because we're always introducing LOXP site into intronic sequence versus exonic sequence, so it's okay to disrupt that sequence. Here, this point, again, when you're making your design, think about genotyping strategy. You can add a unique restriction site in the intronic sequence. This will help you identify and screen for this LOXP site in the future. And because I've talked about kind of the idea of how to make the donor template, if both guides recognize the same strand, then the donor template, of course, will be reverse complement, as I discussed earlier, to the PAM strand sequence. However, because we use two different guides, we have had projects where the PAMs, there are two different guides, they're on opposing strands. If they're on different strands, the donor can be on either strand. For the large donor template, we order these as long SSODNs or plasmids. These are the companies that we've used all very successfully. It's important to know for the large donor template, if you're making a large SSODN, you only have about 100 or 200 base pairs for the homology arms. Of course, as I was speaking earlier, there's a little bit less flexibility, while we use larger homology arms if we're using some kind of double-stranded donor template. Again, I don't know if you want to go through all the little details. Here's some examples. I know this is being recorded. This is a lot of information, but at least it will give you an idea for you can see maybe in this last picture here is where when we have the guide RNA, and this is what I'm describing is that we use the LOXP site to split the guide RNA, and this is how we prevent this being retargeted by the CRISPR-Cas9 when this gets introduced properly. So as you can see, yeah, we have where we're introducing them separately, and this is one long donor template. So for the large insertions, they're kind of following the same best practices we've already established earlier. Again, we're splitting the guide RNA sequence or introducing silent mutations in the PAM or spacer sequence to make sure that our donor template doesn't get cut by the CRISPR-Cas9. And again, you can order these donor templates from the companies that I've listed before. I put an example, as I've said, this is one of our larger constructs, just to give you an idea of how big a donor you can put. We have never, I think, exceeded 5 kb, but I do have labs that have reported up to 9 and even 14 kb inserts. So there's a lot of possibility out there for you. As you can see, this is Cree ER-T2, Cree ER-T2 EGFP. And we actually did this quite successfully in I think, the first or second round of microinjection. They got all their founders at about a rate of about 20%. So now that I've talked about choosing the guides, validating guides, you have a donor plate, you're like, Mitra, I've done all my work. Here are all my components. Deliver them, make me a mouse. So when you come to our lab, we have a variety of ways of delivering actually the CRISPR components. And so we can actually deliver them either through microinjection or electroporation of zygotes, or we use a method called iGonad. So basically how this works is that for zygote microinjection or electroporation, what we do is we collect zygotes from females that have been mated to stud males. And these embryos are collected, and then as you can see here is we will directly introduce the CRISPR-Cas9 components through microinjection, or through electroporation. We will either do embryo transfer immediately or culture them overnight, and they will be all these embryos that have been manipulated will be transferred into what we call a pseudo-pregnant female. These pseudo-pregnant females are females that have been mated to vasectomized males. And so we know exactly where they are in the state of estrus. And then what we do is we reintroduce the manipulated embryos back into the oviduct of these pseudo-pregnant females, with the goal that we will have pregnant females that will deliver pups. I want to talk about iGonad, which is a procedure that actually Yojiro has helped us establish. So we're excited and we're hoping to be able to expand our ability to do this. iGonad allows us to actually excise the ovary with the ampulla section where we will know on the day of the plug where the embryos are actually located. And the idea is that we would deliver the CRISPR-Cas9 components next to the fertilized embryos, and then we will do the electroporation on the ovary, excuse me, not the ovary, but on the ampulla directly. And then we will close up that female, and then that way she will deliver actually pups that have been, that are genetically mutated. This is a protocol that we're developing. It works with some efficiency, but it's not quite yet as well established as our microinjection and our electroporation techniques. Although we do use it, we haven't used it as much. We're quite excited about this technique because it allows us to reduce the number of mice we use and to do less manipulation to embryos, where here we actually have to do matings, sacrifice females, collect the embryos, do the manipulation, and then return the embryos back into a pseudo-pregnant female. Here we would be able to do it directly all onto the female of the strain of choice. What helps us decide is that microinjection and electroporation are pretty flexible and allow us to pretty much generate all our genetically engineered models, from NHEJ to all HDR models that we want, both with small templates and large donor templates. iGonad at this stage is only working when we're working with NHEJ or the small donor templates. So the things that can affect what delivery method we choose is, the size of the donor template. The larger donor templates still have to be done at microinjection, and we actually oftentimes do a lot of injections at 2-cell stage. We could also have to decide how we're going to deliver it, because if we have embryonic lethality, as well as the background strain of the mouse. And then, so what I thought, since we could not actually do any on-site microinjection, is here I can show you a picture of what it's like of Nobuko actually doing some microinjection. I will show you some microinjections. I will show you a short video. Here is microinjections at the 2-cell stage, and this is what we use to deliver our large donor templates. And here you can see is we insert the needle, it's been delivered on one side, and then we'll have to turn the embryo to the other side, and then we will microinject the second blastomere. So I'm hoping someday, post-COVID, some of you guys will get a chance to try that out, and we will be able to show you actually all of the delivery methods for us. So now we've delivered it, pups are born. What do you have to do? You have to screen your founder mice. That in itself is complex, and now all that work you've put into the design and thinking about your genotyping strategy comes to play here. I have to stress, and there's many reports about this, when we deliver the guide Cas9 RMP complex, these F0 mice that are born often carry more than 2 different types of alleles. These are mosaic mice, and that is because the CRISPR-Cas9 can cut several times through early development because we're delivering the components at such an early stage of development at 1 cell and 2 cell embryos. So because of this mosaicism, genotyping can be complex. It is sometimes hard to identify all the alleles, and sometimes it is hard to actually have your strategy work. For this reason, even though you might have beautiful Sanger sequencing on your F0 mice, I highly suggest and kind of would wish that everybody will do a final confirmation on the F1 generation, and then to be sure that everything, all the mutations are there present as you desired, as well as no other carrier mutation. Even though the mutant mice, the F0 mice that are born, are mosaic, the advantage that we have working with animals versus people who are working with cells is that the alleles will segregate. Even if you can have 6 different mutations in this mouse, because the alleles will segregate when we mate these F0 mice with a wild type mouse, each allele will segregate and be transmitted individually from the parent. This allows us to do actually very accurate genotyping and confirm the germline transmission. So screening can vary depending on how you want to try to detect the mutation. Whether you're trying to do kind of a small mutation, that's usually done with kind of the same genomic PCR strategy that you develop for guides, for the guide screening. So this is why it's worth taking all that time and trouble to do it in the beginning, because you know it will work, is that primers, of course, are chosen outside of the donor template. And then we kind of use the same strategies that we did for screening for guide validation, and everything is sent for Sanger sequencing. And whoops. And then the goal is to be able to align your sequences, as you can see, and then identify your sequences. And here, this is the chromatogram. When I showed you that earlier design where the researcher was asking us to delete these 2 alleles, and you can see this is the chromatogram of a beautiful het, and as you can see, as the sequence gets shifted. So you really, this is also the reason why we say it's very important to be able to look at the chromatogram, so you can look at the different peaks, because you can see that you will see multiple peaks, and these will be able to help you identify the mutations that you have incorporated. It's fairly, as I said, you use kind of the same strategy that you would use for the guide testing. This is the place where you can use a restriction site to help with your genotyping if you thought about it ahead of time in your design and you incorporate it actually in your donor template. And then now I want to talk about screening for the large insertions, large donor templates, which include both the conditional and the large knock-ins. The conditional alleles, they're very challenging to screen because you want to do a short PCR to actually screen and detect, I apologize for that, oops. Screen and detect for the individual LOXP sites. I apologize. Ugh. As well as screened for the LOXP sites being in cis versus trans. I spoke about this earlier. This is a nice little figure that I have from Yojiro explaining it, is if you have the alleles inserted in cis, as you said, when you mate to wild type mouse, you know, you're going to be able to identify it. If they're in trans, they're going to segregate, and this is not a useful mouse whatsoever, okay? There are some challenges. You can try using a long range PCR to PCR across both the LOXP sites, depending how far apart they are. It will kind of determine how likelihood your PCR is going to work. We have worked in developing an in vitro Cree assay to kind of see if we can actually see the Cree assay work on the 2 LOXP sites to detect a deletion in the founder mice. But no matter what, I would screen in all F1 mice that the LOXP sites are in cis. and that the integrity of the LOXP sites are there. And the reason I'm saying this is because there are a number of researchers who believe that the LOXP sites are in cis and they're correct, but afterwards they find that there's a small mutation in the LOXP sites. LOXP sites are palindromic sequences, and they sometimes will be missing a single LOXP site, or they'll insert an extra, they will be missing a single base pair, or they'll have an addition of a base pair. This means this LOXP site is not correct, and even if they're in cis, they will still not work. I just talked to a researcher who discovered this after 2 generations when they were mating to Cree mice. They finally rechecked it, and they found out that one of the LOXP sites was missing a single base pair inside of it, so the Cree mice were not working and they couldn't get the excision of interest.
Excuse me, Mitra, you have about 5 minutes left.
Perfect. I'll wrap it up. Screening for the large insertions. Basically, how we do this is because they're mosaic animals, we'll use internal primers just to look for the donor template that you wanted to insert, something like a GFP or an RFP. And then what we want to do is external primers to verify the junctions for the correct insertion at both the 5' and 3' ends. However, because as I mentioned, mosaicism can make this challenging, oftentimes that researchers will identify with internal primers. Let's say if they have 10 mice, they find 3 mice that are potential founders, they'll breed those to F1s, and they'll be able to actually confirm with external primers their correct insertion in the F1 mice. So now that I've kind of given you the whole gamut, given you a whole course, how to make your CRISPR mouse, I want to talk about some genome engineering applications that we've done with researchers here. Here I want to introduce, actually, this is an NHEJ project that we did with Maxime Bouchard's lab. And why I like to use this as a good example is, even though I steer most people through homologous recombination, here's a good example of why NHEJ project worked really well, because we were looking to actually create double knockouts. And interestingly enough, Dr. Sanchez, orally in Dr. Maxime Bouchard's lab, she wanted to target 2 different locus, and actually she wanted to target it in a mouse that she had already had inside of the lab that was already pre-targeted. It's a PAX2 GFP. So what we did is we designed guides to delete critical exons in the 2 different locuses, as you can see. And because we really wanted to create knockouts, we designed 4 guides for each locus. So we injected a total of 8 guides at the same time. And then she then collected embryos at E9.5 and E10.5 to look at the different genotypes, and she was able to collect enough of either single genotype or compound heterozygotes, and she also has some compound homozygotes, so she was able to really show some nice defects in the nephric duct and kidney development. So this is really something that you can do with CRISPR-Cas9, and this shows you like the true flexibility of the system, is that we were able to target multiple locuses in a mouse line that actually already she existed in her own lab, so that she was able to actually screen and see if these 2 targets were the critical targets or not, to generate the phenotype of interest. So then, as I've talked before, is we've done also for Dr. Muller, and this is another reason why I like so much working at the Cancer Center, is that he had a point mutation line that he had created by embryonic stem cells, which I forget how long he told me took him, but quite a while. And within, I believe, 5 or 6 months, we were able to take his original ESR1YS model, which he's published upon, and then we actually changed the original mutation and introduced a new conditional point mutation that he wanted within about 5 or 6 months. So he was able to generate a new point mutation model without going through all the pain and suffering of generating these LOXP sites. So then the last thing, since we are short on time, I just want to talk about is that breeding and expanding your F0 mice. There are a few key points I really want to stress. As I said, these mice are always mosaic. Always breed your wild type mouse purchased from a vendor. We have one of our speakers later on, and I'm going to make your pitch that they need to be your best friend. I will be happy to provide you with genetically engineered mice, but you do need to actually use mice generated for vendors so you don't have any other carrier mutation. All your founder lines need to be bred individually. And I really, again, I've said this several times, your F1 mice should be carefully regenotyped, resequence them, please. If you have not developed a long-term genotyping strategy, you really need to kind of analyze it now. And we do generate a large number of founders. Not everybody wants to carry all these founder lines. This makes sense. It's expensive. It's time-consuming. You can cryopreserve lines to reduce your founder lines. So now, just to wrap it up, this is kind of the McGill Integrated Core or MICAM facility. I will have to say that the microinjection setup and a lot of all that CRISPR-Cas9 technology was developed with Yojiro, and he really put everything in place. And there's me, and there's Nobuko Yamanaka. She's our key microinjectionist. Shad Desjardins does all our CRISPR work. She'll work with you for designing everything. Marie-Pierre Cloutier has joined us, and she's responsible for doing a lot of our surgeries and taking care of the animal colonies and making sure that the mice are healthy and happy, which is kind of the key step in making sure our facility functions smoothly. So I'll then, if there's any questions, or we can move on.
Okay, thank you, Mitra. There was a question very early on, but I think it's appropriate you answer it now. You mentioned that most modifications can also be done in rat models. Have you successfully developed lock screen models of rats?
So lock screen models of rats, all right, yeah. So I have never done it. And actually, interestingly, at the last transgenic meeting, I spoke to there's a small group of us who work with rats. There are not a lot of people. The popularity is always with mouse models, but there is definitely interest and drive. I think the challenge with the rats is the rat lines, they tend to be more outbred, so they are more challenging to make. I have personally not done it, but there are other groups. The Wisconsin group, who has developed a lot of the engineered nucleases with zinc fingers and talons, they have had some success, and there are several groups in Japan who have done it. So there was discussion that we needed to kind of make our own little rat group to share best practices to be able to generate conditional rat models. I think that is actually more viable because we can use small donor templates. Where we have, well, not me personally, but where they have struggled more is actually making the large knock-ins. They have not made, there have not been a lot of large knock-ins made using CRISPR-Cas9 in the rat models at this stage of the game.
There are no other questions in the chat. Bill Muller is back. Yojiro is also on the Zoom. If anyone has a question, I think, Thomas, you have a question.
Yeah, let me try to ask this before. So there's a lot of diversity in the enzymes that are allow you to edit genes nowadays. There's all sorts of flavors and colors. Way beyond Cas9, and it seems like we're locked in, and as it is very often with those technologies, locked in the first version of things. So, for example, a lot of the limitations are due to that PAM. You need to have that PAM near what you want to modify, whereas there's other enzymes that allow you to engineer and target sequences, let's say, that are AUH and have those GCCs around. So could you enlighten us in terms of, availabilities, in terms of tools, diversity of tools? Do you have any variants of Cas9 that loosen the PAM, for example, and or as I think you answered before, different types of enzymes that can be used to engineer? Which one are available in your core, and are you contemplating, are comfortable working with other variants?
So we have used CPF1, which is one of the variants, and we have used that successfully. We have targeted with CPF1, and I think we have another project coming up with that. Yes, there are definitely other variants. Actually, I think what I think would be interesting to work with is using base editors or kind of prime editing. Those are kind of the newest developments of CRISPR-Cas9, because they don't need the PAM quite the same way. You have a little bit more variation. I think there was hesitation because that technology was still being developed. But yes, I mean, right now we're open to certainly using any of these types of different technologies.
Yeah, so some of those, like you mentioned the base editing, I'm glad you're mentioning it, but it allows you to engineer different kind of lesions also, right? You might have a greater diversity of options in terms of lesions that you can engineer. Let's say if you want to screen or paint, let's say, a domain into multiple variants, and so there are options that would bring you sub-molecular resolution, sub-loci resolution with some of those tools. Okay, but what you're saying is that they're still being tested and not sure that they're trustable, right? Because there's always the off-target possibility, right? When you remove the PAM, there's a chance that this thing starts shooting around everywhere in the...
Yes, but actually, I would say I think now the technology has been developed enough that it's worth establishing now and trying it. It's the problem as a platform for me is that when people come with a project, they want their mouse model as quickly as possible and within their budget. And so the problem with using new technology is that I can offer it and I can give you the pros and cons, but usually it takes a little while to establish it and you have to make a few mistakes to figure out how to do it.
Yeah.
Have best
development, time, and risk, right?
Yes. So the question is, are you willing to be the project that has the risk and the time and the money that's involved in that? And I think that's always a risk. But I would say that that's the flexibility that it's allowed here, is that we definitely have developed a lot of these technologies and been able to do that because, people are interested in using these more, the newer technologies that are available, but,
So there's another question in the chat that just popped up from Marc Sabelel. Very nice talk. Thank you, Mitra Cowan. I was wondering if you checked by Sanger sequencing your off targets for each guide RNA. Thank you.
So we won't check it ahead of time because the goal is to have pre-check the best guide RNAs ahead of time that have the lowest off targets. So there's a few strategies to handle off targets in mouse models. You can certainly take the top 5 hits that CRISPR will give you of saying what are the off targets. If you look at that earlier slide that I presented, it's in one of it said 236 off targets. So you can pull down that list and see what are the lists, and you can see where are the off targets going to be. Are they going to be in exonic sequences? Are they going to be intronic sequences? Are they in critical genes? And then you can actually, by PCR, amplify those regions, look for and send it for sequencing and seeing if there are any off targets. I certainly have explained this to researchers. We don't do that ourselves. We actually give the information to the researchers. I've only had 2 PIs actually do that, and they have found it. They have found off targets. The other option is what most other researchers do, and that is to continue to breed out cross or back cross to wild type mice. And what I mean is by taking your founder mouse, breeding it to wild type mouse, having F1, taking the F1, breeding it to wild type mouse, and then keep doing that for several generations. If you do that, you're going to breed out any off targets. The likelihood of carrying any carrier mutations, unless they're very, very close to your targeted mutation, is, you know, zero. So that is what I recommend all researchers to do, and I think most researchers do that. And the last way to deal with that is our goal is always to produce multiple founders for every project. That's, you should test and look for your phenotype in all founder lines. If all of them are reproducing the phenotype of interest, then it's not, then the likelihood that it's a function of the CRISPR-Cas9 has caused some kind of off-target carrier mutation that is generating the phenotype that you're seeing versus the targeted mutation is pretty much none too, you know. So the goal is also to have multiple founder lines, and that's what everybody should be trying to do.
Are there other questions for Yujiro or Bill who are still online? You can unmute if you want to ask a question. If not, I wish to thank the three speakers of this morning who have done an amazing job at presenting the work they've done for sometimes decades. Most have done decades of work. And I hope it's been very instructive to those of you who are contemplating developing new mouse models. It's a long process, so you need to walk through it very closely and very efficiently. And for sure, if you need help with some of these technologies, the MICAM core, Yujiro and Mitra are definitely there to help. We will put the email addresses of the three people who presented this morning, if ever you want to contact them privately for questions, or if you're interested in working with them on a certain project. It is 12:13. We will break for lunch and we will reconvene, let's say maybe 1:10, 1:15 for the next presentation. Please open your windows and breathe something like air coming from outside. And first thing you do, go wash your hands. See you later. We will shut down the Zoom during the lunchtime and we'll reopen around 1:05 or something. Thank you.